osmolarity and the concentration when you did the osmolarity were equal. That would be isotonic. Okay. What you're really looking at for tonicity is which solution has the most salt. Remember, for tonicity, you're looking at salt. For osmosis, you're looking at the movement of water. All right? Make sure the units for each solution are the same. Convert if you need to. Other questions that we might ask you. Again, we go to the osmosis. Where does the water move to? So if I ask you to draw me a picture, you should be able to draw me a picture and make an arrow as to which way the water is going to move.
Low concentration.
To go from high to low. To predict or draw the effect of a solution on a cell. So remember that the isotonic condition for a cell is 0.85%. Anything above or below that, and you're going to be moving water either into or out of the cell. You should be able to draw me that. What happens if you put a cell into hypertonic solution? Use your words.
It shrivels.
Shrivel, what's the big word?
Crenate.
Crenate. Okay?
Shrivels.
What happens if you put it into a hypotonic solution?
It swells and it
Lyses. Big words. Lyse.
I was looking for it.
That's okay, you got it. Explain osmolarity. Why will a molecule of salt break down into a mole of each component atom? What's the bonding structure? What's the bonding structure of a salt? Ionic. Ionic means that it will break apart into its two component parts when you put it in water. Why? Polar. Okay. Do all molecules break apart in a solution?
No.
No. Which one wouldn't?
Covalent.
Non-polar, or give me an example.
Metal.
Glucose. Why? Because they're covalent bonds. It takes energy to break those bonds, not so much for an ionic bond. This goes into the strength of your bonding structures. And this is a big question. Can you think of more questions about these concepts? You should be thinking of some right now. Look at your labs. It is your chance to ask me.
What exactly is the question?
I don't know because I don't write the questions. I only grade them with a key. I have a little bit of input, but not much.
Well, for this right here, are we gonna have any slides showing these kind of reactions, the hypertonic and hypotonic
Wouldn't be a
reactions?
slide. Remember you had dialysis bags?
Yes.
Okay. She could have one of those out.
Okay.
Or she could have a picture of it.
So
Okay,
like,
and tell you that it's such and such a concentration inside the bag. Where is water going to move?
okay.
Yeah.
Do we have this PowerPoint?
No. You need to write.
Yes, ma'am.
You just send it to me.
Any other questions?
I'll give it to you.
This is your chance. You can email me if you think of some. Cell surface receptors. Be able to use the table on 207 to identify a lectin. These are the tables that are going to be on your benches for you. Second page. You need to know what the specific sugar is on the table. Be able to identify the conditions for a hemagglutination or peroxidase reaction to occur, which means you should know what your positive control is, be able to distinguish it from your negative control. You should also know where you get inhibition. So, if we go back to your example of the lectin with sialic acid, where would you see, describe the condition you would see inhibition?
The one with the sialic acid in it.
And you need to put all of those down. Right, but what's the condition? What's on the slide? Condition means what's on the slide.
Blood cells.
The blood
The
cells.
blood cells are not together.
No. What's the condition? What's on the slide? What reagents did you put on that slide?
The limus buffer, the
Right.
polyphemus, and the sialic acid,
Sialic acid
sialic acid, and red blood cells.
and red blood cells. Or cheek cells. If you put cheek cells down, then you got peroxidase and a whole bunch of other stuff that's listed as a condition. All right? So you need to be able to tell me when inhibition occurs. You need to also be able to explain what is inhibition. Can you put that into words?
Inhibition is basically making, forcibly keeping the red blood cells from grouping together.
Not so much, almost.
So they're competing for the same.
Instead of binding to the lectin,
They find the outside.
it binds to the outside sugar, thereby preventing reactions between cells. Okay. Remember, you're binding lectin, not binding cells. Be able to explain osmosis and how to quantify it. That's your osmolarity. Okay. Be able to explain osmotic pressure as opposed to hydrostatic pressure. What's the difference between the two?
Osmotic pressure, the difference in the tonicity of two solutions versus hydrostatic pressure, which is the water pressure from gravity, pretty much, right? It's just
Pretty much, where you find hydrostatic pressure a lot is in blood vessels. So what's moving the blood through the blood
okay,
blood
so just
vessels?
the water, the
Right.
pressure from the pumping, whatever.
The PSI of your
whatever
heart.
pressure is being affected.
Be able to explain or diagram the osmosis feedback loop. Ooh. So go back and look at what hormones were involved.
So that's going to be the water retention.
What's the name of that hormone? He just told us. Begins with a V.
Vasopressin.
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 Where does it act?
Acts in
The kidneys.
kidneys.
Okay.
Comes from the pituitary gland.
Comes from which part of the pituitary gland?
Anterior.
Posterior. There's only two for the posterior, vasopressin and oxytocin. All the rest of those come from the anterior pituitary. All right? Let's see, be able to label and explain the function of the cell membrane structures. So could she give you that picture of a cell membrane and tell you to label it? Yeah, she could. She could ask you, what are the functions? What's the function of cholesterol? What does it do for the cell membrane?
Prevents water from leaking through the membrane.
It actually increases the fluidity of the membrane. It keeps those tails from interlocking and becoming rigid. That's why you find more cholesterol in deep sea mammals, because they go into really, really cold water. If we did that, our cell membranes would become very brittle. Theirs don't.
Freeze to death.
Can you think of any more questions about these concepts? Osmolarity, inhibition, osmotic pressure.
On the cell membrane, could she ask about the potassium-sodium pump?
No, that's a lecture question.
That's a lecture question.
We didn't do that in lab. So if we didn't do it in lab, it's not fair game.
Thank goodness.
Anything else? Okay. So start an inventory for yourself. Do I know these things? Can I sit there and write them out on an exam? Your histology review. You should be able to take that table and compare and contrast the functions of specific tissues for the four major tissue types. What are the four major tissue types?
Connective tissue.
Connective, epithelial, muscle, nervous. She's just kidding. All right? If she asks for a major tissue type, those are your four answers. Don't be giving her stratified squamous epithelium. Don't be giving her smooth muscle. She just wants the major. Explain the purpose of a tissue within an organ or an organ system. So epithelium, she chose to give you a whole lot of functions.
Protection.
Protection,
Secretion.
secretion,
Filtration.
filtration. You need to know all the functions and where they occur. You can see that on a microscope slide. She can give you a slide of, say, the kidney, Bowman's capsule. Have you looked at the Bowman's capsule?
Simple
You
squamous.
should look at the Bowman's capsule. What is the tissue underneath the pointer? If it's pointing to simple squamous, what is its function?
Filtration.
Filtration. Defiltration. Just filtration. Diffusion comes other places. But for the Bowman's capsule, it's the only place in the body it occurs. Right?
Filtration.
Filtration.
Be able to identify the locations of specific tissue types and identify their functions within an organ or an organ system. Any other questions? Okay, make sure that you can read my writing on your charts. If you can't read the writing, let me know now. I can.
We just need to study this for that